Welcome everyone. The lecture will be recorded. So please mute yourself to reduce any background noises. So if you have any question, you can type into the chat box and Dr. Simmerling will answer questions after his lecture. And also taking this opportunity, I just want to thank you to our interim dean, Safid Fasal, my supervisor, Janet, our social media team, and also my colleague, Christine Finner, to provide all the support to make this event happen. Of course, Dr. Simmerling too. The University Library is honored to have Dr. Carlos Simmerling share with us today his important work on finding treatments for coronaviruses. Dr. Simmerling obtained his bachelor degree in 1991 and PhD 1994 in chemistry at the University of Illinois at Chicago, performing early research on methods for computer modeling of biomolecules such as proteins. He then went to a postdoctoral fellowship in pharmaceutical chemistry at UCSF, where he became a lead developer of the AMBER biomolecular simulation software that is used in thousands of research labs worldwide. In 1998, Dr. Simmerling joined the chemistry department at Stony Brook University, where he is currently a professor, and he became the associate director of SBU's Laufer Center for Physical and Quantitative Biology. His NIH and NSF funded research focuses on development of improved simulation methods and models, and using those tools to study biomolecular recognition mechanisms. His articles on improving the physics used in biomolecular modeling have been cited nearly 10,000 times. Dr. Simmerling is currently the Marshall Laufer Chair of Physical and Quantitative Biology at Stony Brook University and a Fellow of American Chemical Society. Now I'm going to turn this over to Dr. Simmerling. Please give a round of applause to our professor. Thank you.
Thank you, Clara, for the nice introduction and for the invitation today. And thanks to all of you for coming to my talk. I know that we all have lots of other things going on right now during the pandemic, and I appreciate you taking time out to listen to what's kind of a story in progress. I want to start out by telling you that I don't have any message at the end about how the pandemic is over, or even that we really know a lot more than we started. But I thought a lot of people on our campus might be interested to see how we use computer simulations to try to help us understand more about the coronavirus and what kinds of things we might do. And so this is a picture to start out with of a diagram of the virus. I'm going to refer to the virus a lot by SARS-CoV-2. This is the name of the virus that causes COVID-19. There are lots of coronaviruses like SARS-CoV-2, and they cause other diseases in humans like SARS and MERS and other things. So there are many coronaviruses. They look very similar to this. And you can see from this cutaway view that they're covered by these spikes on the surface. They're in purple. And those spikes make them look like a crown, which is why they're called coronaviruses. They have other components like their RNA that spiral up in the center. They use RNA as their genetic material, unlike the rest of us that use DNA. Most organisms use DNA, but lots of viruses use RNA. And so this is what the virus looks like. And here's a diagram of the life cycle of the virus. First, it starts out outside the human cell, and then it has to get into the cell. What it wants to do is get its RNA into your cells so that that RNA, once it's released, can be turned into proteins. This is a messenger RNA, and so it gets turned into proteins. There are copies made of the RNA, and then it goes through this whole series of events where it eventually assembles new viral particles that are released, and then it goes through that process again. And so in order to stop or treat patients that have COVID-19, there are really two things that you can do. You can try to attack things that are on the virus side. You can keep it from getting into the host cells, or once it gets in, you can keep it from making more virus. Things like remdesivir try to block the replication of the RNA to keep it from making more virus. I'm going to talk more about how to keep it from entering cells. And then there are things you can do on the host side too. You can make the host cell very unfavorable to the virus so that it's not a good place for it to replicate. And then you can try to treat symptoms and minimize the damage from the infection. But I'm going to talk about that viral entry. How does the virus first get into your cells to put its RNA there and start this process going? So that job of getting into the cells is the function of the spike protein. The spike protein is something that we see on lots of viruses. It's a viral fusion protein. Viruses in general have this task of needing to get their genetic material into your cells. That means it needs to get through the cell membrane. And so what it does is it's going to fuse its membrane, which is down here in the bottom in these yellow blocks, with your cell's membrane, which is the yellow blocks that are up in the top. And then this is a time sequence, little cartoon, showing you as it proceeds to the right, it's pulling these membranes together and eventually fusing them so the virus gets into the cell. And the way that the spike does that, the spike is down here in the bottom, is that the spike is going to interact with another protein that's on the surface of the host cells that's called ACE2, for angiotensin converting enzyme, and that's involved in regulating blood pressure. So the spike is going to undergo some motions that we'll talk about a lot. Eventually it ends up touching the ACE2, and then it goes through another series of large changes and attaches itself to the host cell surface, pulls them together, and then ends up going through the fusion. So this story is really about two proteins. It's about the spike glycoprotein, and it's about the ACE2 protein. So some of you probably aren't biologists, so you're probably asking yourself right now, he's talking about proteins, and I don't know what proteins are. So we all know proteins, other than things that you need to have in your diet, like a protein shake or protein energy bars. Proteins are sort of the workhorses of biology, and they do lots of different functions that I've listed here over on the right. These are just a few of many of them. They store things, they move things around, they carry out chemistry, they're involved in your immune system, and they're parts of viruses. So many, many different things. This image on the bottom left is a protein called myoglobin. It's involved in storing oxygen in your muscles. Now, where do these proteins come from? Well, it's got this sort of complicated three-dimensional shape that I've drawn here as a ribbon. We do that often because the chemistry shapes are very complicated. These are just big molecules. And so I'm going to be drawing with these ribbons a lot so you can see sort of the overall shape of the protein. But actually, they start out as these very flexible chains of amino acids. And this is why you need proteins in your diet, because you need amino acids to be able to make proteins. How do we know which amino acids to put in the proteins? That's the job of your DNA. And so your DNA tells the code that turns into RNA, and that RNA by ribosomes gets turned into proteins, and then they fold up and they have a shape and they have a function. So that's introductory biology all in one slide. So we're going to be talking about proteins. Now we can see these proteins in experiments. I showed you that cartoon in the beginning, but this is some really exciting recent work where these are images of actual viral particles. And you can see they've got kind of a circle shape like you saw in the beginning. They have their own membrane, which makes them a little bit delicate as viruses go. And then they have these protrusions on the surface. They're the spike proteins, and we can just barely get a view of what these look like. Now, the reason this is so exciting is because these viruses are really, really small. They're about 100 nanometers in size. And 100 nanometers out of context doesn't mean much, but the width of human hair is about 100,000 nanometers. And so this is 1,000 times smaller than a human hair. It's really, really tiny. So the fact that we can see them by experiments at all is really amazing. Now, they're very small particles, but we can keep them from getting in us. And this is one of the reasons that we wear masks. And so this is a picture of an N95 mask. And N95 just means that it filters about 95% of particles that are 300 nanometers. That's the measure that they use. Now, it seems like the virus would still be able to get through that, but the viruses don't travel around on their own. Like I said, they're pretty delicate, and they're usually in clusters, droplets of water and mucus and things like that. So the particles are bigger than that, and the N95 masks and other masks are pretty effective at keeping them out. So these are small viral particles, and we can see what they look like. Now, we can also get much more detailed views of what these look like when they're not on the virus. And so this is what the spike looks like in one of those ribbon diagrams. And there's lots of different parts to this. So I'm going to point out a few of them because they'll come up a little bit in my talk. The first thing is that I'm showing you one spike protein here. They actually come in clusters of 3. It's called a trimer. 3 pieces fit together. And there's different parts of it. The one I'll talk about a lot is called the RBD, the receptor binding domain. That's the part that's going to interact with ACE2. And that's kind of like the key that fits into the locked open entry into the cell. And the RBM here is just part of that that actually touches the ACE2. There's some other domains here. And when you look at this, it's just all one protein. But in reality, it's not one protein. There's something interesting about this protein, and that's that after it gets made, it gets cut into pieces, and that's very important for its function. And so this is just the length of the protein showing you kind of like a string here. And during processing, this protein gets cut like by molecular scissors in two places. And so I'll talk about those a little bit more later. So when you look at this protein, I'm going to rotate it so you get a better view of it. It's a pretty complicated structure. The viral surface is going to be down here at the bottom. It's kind of a stalk. And now I'm showing you the whole trimer. And so this is really what that spike protein looks like. It's pretty big and complicated and hard to follow quite what's going on. And so I'm going to show simpler versions of this most of the time. This one's showing you a space filling version. So you see just a little bit more of the shape of the protein. One of the interesting things is that this version of the protein, these parts that are at the top, these RBDs that are in the red, yellow, and blue, the three different versions, this is what we call the closed structure. And it's closed because these RBDs are all kind of touching each other. And so one of the things you can do is say, well, how would this interact with ACE2? And you can try to put the ACE2 on it. We know where it should attach. And you find that if you try to do that, it doesn't work very well. This gray ACE2 molecule ends up overlapping the other monomers, and it just won't be able to fit in. And so that's not how this works. We actually have to have another structure for this that's called an open form or an up form, where this RBD has moved up, and now it's more exposed, and now we can put that ACE2 on and see very clearly that it would fit and that interaction would be able to happen. And so the closed form isn't able to bind to ACE2, and it's not able to unlock entry into the cell. And I'll come back to that a couple of times. So that open RBD, it turns out, is so important to getting into the cell that that's also the target of your immune system. You eventually will develop immunity by developing antibodies, which are other proteins, and I've shown those here in these gray ribbons. These are 10 different experimental structures where people have been able to find out just where that antibody binds, and they bind to the spike, and they bind very close to where the ACE2 binds, and so they can block the ACE2 from binding. When the antibody binds to the spike, the ACE2 can't get in, and this can't do its job, and so they're very good at blocking it once you develop immunity. Okay, one way of developing immunity other than to actually get COVID-19 would be to have a vaccine. We don't have vaccines yet, but there are quite a few of them, 145 right now, that are in development and about 35 in clinical trials. This is developing vaccines on an unprecedented scale. We've never done anything this fast in science before. And so it's good that there are lots of different variations on this. Some of them are trying to deliver RNA into your cells. Some of them are trying to deliver DNA into your cells. Some of them are trying to deliver spike proteins, but eventually they're all about the spike. All of these vaccines want to expose your immune system to the spike so that if you do see a viral particle with a spike on it, your immune system is already ready to go and bind to it and stop it, and you don't get sick in the meantime. And so that will be really good once these are ready. And once we figure out which variation on this works, and many of these types of vaccines, there is no vaccine that's been developed using this kind of technology, so they're all very new. Okay, well, the vaccines should be great, but one problem with vaccines is that they bind to that receptor binding domain that I pointed out. And as with many things, once we get immunity to one version, we don't necessarily have immunity to another. And so in this animation on the right, I'm showing you that spike surface color-coded by how much that surface varies between different coronaviruses. So SARS, MERS, all kinds of different coronaviruses that the gene sequences are known, and we can map that to the surface. And we know that the antibodies bind up here, but it's very red. They're very variable, which means that a vaccine may help quite a bit against COVID-19, but having that vaccine and that immunity won't necessarily help us during the next coronavirus that comes. And we've had them before, and they probably will keep coming. And so this is really a challenge, is how can we find something that might be a little bit better target, something that the next coronavirus is going to look the same, so we don't have to go through all of this again and develop new vaccines. And so that's really a challenge for us. So what I'm going to mostly talk about today isn't on the vaccine side of treatment, which is the most likely thing to help us for COVID-19, but it's about how are we planning now for the next coronavirus pandemic, and what can we do so that we're ready to go and not have to have all of this slow vaccine development. And so what we're going to do is we're going to look not at the surface of the molecule that varies a little bit. We're going to look down at some of these parts down below that are in the white and blue regions where it doesn't change between different coronaviruses. And we want to know, why don't these things change? Why is every coronavirus the same there? And what can we do about that? And so we're going to target function and not surface or shape. So a good way to think about this is to think about cars. Let's think that you're an environmentalist and cars cause pollution, and you decide that you want to interfere with cars, just like we're trying to interfere with coronaviruses. Now you look at a few cars and you say, well, all I get to see really from these experimental structures is I get to see what the shape looks like, what the color looks like, the surface, and so on. And these different cars, they're different colors, they're different shapes. This one here, this Tesla, it doesn't even have a gas engine, it's an electric. And so it's really hard to look at these and say, given what I know about the surface, coloring and shape and so on, how would I find something that interferes with all of these? But that's not hard for you, and you're probably already thinking of ideas because you know how cars work. And you know that one of the way all cars work is that they all have doors, right? We understand the function and the moving pieces, and understanding those moving pieces, we know that doors are part of every car. And if we could find ways to make doors so they don't work anymore, then that would stop all cars. And it would be much harder to design cars that don't have doors anymore. And so that's kind of our target here, is to say, can we learn about functional motions so that we know the pieces of the virus that are going to be very hard for it to change because it just won't work right anymore. And that's what we're going to do with the spike, is try to understand what its job is. Okay, so as I mentioned before in my little diagram, this spike is going to interact with this ACE2. So what happens next? That doesn't let it into the cell. So what happens next isn't very clear, and this is the part that we want to try to understand more about. Here's our structure on the left of our big complicated spike trimer. It's bound to an ACE2. Something has to happen. The other experimental point we have is all the way at the end, this structure over here, where we know what it looks like after it's already pulled the membranes together. And the membranes are up in this sort of blobby section that we don't have a clear idea what it looks like, but we know the spike looks like this now. And so if you compare these two, the pre-membrane fusion form and the post-membrane fusion form, they're really different from each other. And so one of the things we want to try to find out is, how does it change from one form to another? And is there a way that we could block those changes from happening? So one thing that we know has to happen is that this orange part here, these S1 subunit parts of our trimer, those have to leave. Somehow after they contact the ACE2, they dissociate. So there's going to be a dissociation or a leaving, or what we call a shedding event of the S1. And then later on, we have to have another cleavage event because we have to release this piece that's going to go and attach itself to the other side. So there are things that have to happen, and we know that based on biochemistry experiments, but we don't have a good idea of what they look like. And structure is one of the key things in order to be able to design things like drugs, because we want to know what's our target shape. And so we think, for example, that there must be some state that looks like what's called this S2 subunit, where the S1 orange subunit is left, and all we have left is this base part. But we don't know what that looks like from the experiments. And then eventually it goes over here. So this is what we're trying to figure out, is what happens along the way. Okay, so a little more about that S1 shedding. This is something that's actually very important for SARS-CoV-2 and COVID-19, because that dissociation event where the S1 comes off is much easier in SARS-CoV-2 than it was for previous viruses. And so up here at the top, I've got the gene sequence of our virus, 30,000 base pairs of RNA. And so the spike is this piece here. We'll zoom out on the spike. We've got an S1 subunit and an S2 subunit that gets turned into a protein. And then if we zoom in right to the part that's our sequence between the S1 and the S2, now I'm showing you a comparison of the protein sequences for several different coronaviruses. And you can see in this spot here, all of these other coronaviruses just have gaps. They don't have anything there. But the SARS-CoV-2 has this little bit of a sequence inserted. It's just a few amino acids. This protein is a tiny bit longer than the other proteins. And that ends up being really important because that is the recognition spot for an enzyme called furin, that as soon as this spike gets made in the cells and the viral particles getting produced, those two pieces are already cut apart. And these other viruses don't have those two pieces cut apart already, and they have to have something extra happen during infection, and that slows things down. And so the genome for SARS-CoV-2 inserted this extra little bit that we didn't see in SARS, and it makes it more infectious because it's pre-primed, it's already cut into the pieces, and that S1 is much easier to take off than it was before. So this early on, and unfortunately recently, was the basis for some conspiracy theories that have suggested that SARS-CoV-2 was actually produced in a lab, because you look at these other sequences and say, well, these didn't have these, and all of a sudden there's a new part. We didn't see this before. That must have been engineered as a bioweapon by somebody. And there are actually papers that people published that try to promote this theory of SARS-CoV-2 as a bioweapon. But it turns out that even though it's rare, it's not unheard of. And if you look at some of the other coronaviruses, even the one that causes MERS, we do see very similar insertions where they have furin cleavage sites, and it happens in the same way. So this has been pretty thoroughly debunked, that it is not something that was created in a lab. But that does make it different from SARS, and it does make it more infectious. But we'll come back to that a little bit. Okay, so here's our diagram. That S1 subunit has to come off. One of the very interesting things that we found from experiments is that that S1 didn't come off until it was at the host cell surface attached to ACE2 for some of the other viruses like SARS. But now, because of that cleavage, it's more infectious, but it also gives it a weakness, in my opinion. That S1 subunit can come off on its own, and experiments have shown that it doesn't need an ACE2 or host cell around. Sometimes it just comes off. It's a little bit of a wobbly piece, and it can fall off on its own. And so there's a recent paper in Science from Bing Chen's lab that showed even though they could see intact spike particles, they also saw some of these post-fusion S2 particles even without an ACE2 around, suggesting that it might happen spontaneously. And they suggested that some of those might be on the spike surface. The reason this is important is because those S2s don't have the ability to bind to ACE2 anymore, and they can't cause membrane fusion. They're just triggered, and they've already done their job, and they're not going to be useful, at least in any way that we understand. Okay, does that happen in actual viruses? It turns out it does. I showed you this picture earlier from this recent Nature paper. They also identified, even though the pictures are fuzzy, they were able to refine them, and they identified some post-fusion states of the spike in the surface. And this is a model they came up with, which is very similar, that there's these spike particles on the surface, but some of them have been triggered prematurely. And so that's one of the things that we're going to focus on. This could be something that's a vulnerability, even though it has made the virus more infectious. If there's a way that we could trigger all of the spike particles instead of just a few of them, then it's possible that it wouldn't be able to infect cells anymore. And so that's a hypothesis that we're exploring. Okay, so overall, what are our opportunities for treatment here or for preventing coronavirus infections? Well, first of all, block ACE2 binding. That's the one I showed you at the beginning. This is what the immune system is trying to do and what vaccines are trying to teach your immune system to do. And that's the step here where we have this binding event to the ACE2. Can we stop that from happening? If we can, it's very effective. The problem is that while you're waiting for your immune system, you can become quite sick, and we know the outcome of that pandemic. Could we do something earlier? Maybe we could keep that RBD from moving back and forth. We know it moves, but if we could block it, then maybe we could stop that process. Maybe we can figure out how that irreversible shedding of the S1 happens. Once that S1 comes off, it doesn't come back. Could we trigger that? Is there a way to get it to happen too early? That could inactivate the viral particles. And then some of these later steps in the process that I'll only talk about really briefly is once that S1 dissociates, how does the virus attach itself to the host cell? How does it pull those host cells together and fuse the membranes? If we had any structural view of that at all, it might be a good target, but we're really missing data there. So what we want to know are not just these snapshots that are in the cartoons that I showed you, but we want to know about these arrows. We want to know what happens during the arrow when we don't really know from experiment. And so that's where the computer modeling comes in. So experiments are really good, outstanding at giving us views of structure, like this protein structure that I'm showing you here in 3D. But those are averages over lots of molecules because they're so small, and averages over time. And one of the weaknesses that we don't get to see all the motion, and things that are moving around tend to be fuzzy and they're missing. And so what we want to have is we want to have a model like this, where we actually see all of those detailed motions. And to quote the great physicist Richard Feynman, everything living things can do can be understood in terms of jiggling and wiggling of atoms. And so that's what we're going to do on the computer. I'm not going to tell you all the details about how we do it, but we are going to put all of our atoms in, and we're going to calculate things like energies and forces, and we're going to have our atoms wiggle around and tell us how these things actually move. Now, the one thing I will tell you is that these calculations are not the kind of thing that you do on your laptop or your desktop or even any computer that you have around. Most of the simulations that I'm going to show you today were done on a computer called Frontera. It's the 8th fastest supercomputer in the world, one of the fastest that's available to the public. Your computer that you work on probably has 2 or maybe 4 CPU cores. This one has 450,000 of them. And it has lots of GPUs, which are kind of like the cards that drive video games. And we use those as well. And so using this big computer, we're able to do our simulations of the virus. Okay, so it takes a big computer and a lot of programming and people. Is it any good? Do we expect to have anything reasonable out of this? I gave a version of this talk recently, and one of the people in the audience said, yes, but how do you know it's any good? Which is really a great question. So are they any good? Well, what do we want to know from our simulations? We want to know a little about structure. And so this picture I'm showing you from a few years ago worked from my lab, where we showed that for a series of different proteins of different shapes and sizes, we could start out just by knowing the protein sequence and in the simulation, get it to fold into the right structure. And so we did a pretty good job on structure, and I think our models are getting reasonable at that. We also want to know about energy. How is the energy of interaction between this protein and other proteins? And so this is an article from my lab last year, where we showed that we could calculate the interaction energy between 2 proteins and then vary the sequence in the protein and say, how does that change the interaction energy? And this is comparing those interaction energy changes between the experiment here on the x-axis and our calculation on the y-axis, and we do a pretty good job of reproducing those energies. And then the last thing we want to know about is motion, and some work from my lab a number of years ago. We had a series of papers where we looked at an HIV drug target, HIV protease, and explored the motions involved in opening and closing of the active site and how drugs like this one, ritonavir, get into the active site and can bind to it. And so we think we have good precedent for being able to do these kinds of calculations, not just my lab, but our entire simulation community. We're starting to do a much better job, and as computers get faster and faster, we're doing a better job of this. But it's important to keep in mind that they're just models. I'm going to show you movies and videos, but they're just models. And this quote is nice. Essentially, all models are wrong, but some are useful. We know our models are wrong. They have lots of limitations, lots of approximations, but we hope they're useful in helping guide experiments and helping understand experiments and helping people decide what to do next. Okay, so that's what we're going to do on the spike. So we want to do this on the spike glycoprotein. So the first thing you might say to yourself is, oh, it's a glycoprotein. What does that mean? Well, they have glycans. And unfortunately, one of the problems is that the glycans aren't showing up in the experiments. There's a lot about the structure that we don't see. What's a glycan? Well, a glycan is something like this, showing you some chemical structure. It's a long branch sugar. You can think about it like the sugar that you have in your diet, sucrose, but it's just bigger and more complicated. And then those are attached to the surface of the protein. So in green here would be an attachment point, and then we've got all these branch chains that stick off of it. And these glycans get added on by the cells after the proteins are produced. We don't really know where those are, and we don't know where some of the surface loops are. But luckily, we do know what those glycans are. And here's an example in a paper from Max Crispin's group where they did mass spec experiments and were able to figure out where and what kinds of glycans were on the surface, and we were able to use that to build models. The other problem here is that this protein is really, really large. It's bigger than anything that my lab has worked on before and bigger than what most people have worked on before. The simulations are going to show you tend to be millions of atoms, and this is why we need these very large computers. Okay, so here's a picture of what our initial model looked like. The blue are those glycans that are on the surface that are sticking out. We've built in all the loops and things like that. This is a collaboration with Rami Amaro's group at UCSD and Vigeli Koutsias at Stony Brook. And once we get that spike model complete, we can start to think about doing simulations of it. We put in water, which I won't show you in most of our simulations, but it is there because water is important. And then we can start to look at what our structure is doing. So the first thing I'm going to show you is this early event that I showed you, that RBD has to move into an up position in order to bind to ACE2. If it's down, it can't bind. And so we can follow that in our simulations and say, how in detail does it move and what do those snapshots look like in between and what kind of motions happen? So we're able to do that. And like I mentioned also, we can map out energies. So I can say, well, what is the energy of this molecule doing? So for those of you that aren't used to looking at things like energy surfaces, this isn't super important. You can think about it like a map showing topography. We've got a couple of valleys, one really deep valley here in blue, and then another valley that's higher up. The deepest valley is the closed structure, and then this one is higher up, and then we've got sort of a mountain passed across, which is why we don't see it much there in the experiment, because that's a high energy state and it just wants to fall down into the valleys. So we can calculate from these free energies that there's maybe about 1% of that in the open state. It's almost always closed, and that's consistent with experiments that show that it's almost always closed. It doesn't open very often. Okay, so that's good, but that doesn't really make sense, does it? It has to open to bind to ACE2. The whole job of this spike is to bind to ACE2 and let itself into your cells. Why would it be closed all the time, right? It should be open a lot, because that's the only time it can bind to ACE2, right? So this is just a reminder what I showed you before. The most efficient thing should be open all the time, but that's not what happens. So why not? Well, in order to understand that, we need to look at these glycans on the surface and think about your immune system as part of this too. This is the down state or the closed form, and the ACE2 is going to bind to this purple part here. The ACE2 can't get in, right? But that ACE2 and the whole receptor binding domain is hidden. It's shielded by these antibodies. These are a bunch of snapshots from our simulations. As it's moving around, this blue part you can see covers that, and that's the spot where those antibodies are going to want to bind. And so they really can't get to it in the closed state. And then in the open state, now this receptor binding domain is exposed and it can bind to the ACE2 receptor. And it's also vulnerable to your immune system. And so the reason it's got this closed to open motion is it's trying to balance being open and being able to recognize ACE2 and bind to it, but also trying to do its best to hide this receptor binding domain from the immune system and keeping it so that even if you have antibodies, most of the time they can't get to it. And so it's that balance that's leading this, especially in SARS-CoV-2, to be hidden almost all the time. And this is hidden more than we saw with SARS. And so that's a challenge, even for vaccines, is that you're trying to bind to a target that is mostly hidden. Okay, well, what if we could counteract that? Is there a way that we can say, SARS-CoV-2 changed a lot about its spike protein to make itself hide more often? Let's fight back. Let's try to make it so that it can't hide it at all. Could we force this to stay exposed? And so this might seem like a bad idea. It has to open to bind to ACE2, so let's help it bind to ACE2 better, but I think that there could be some good things here. One would be if we had some way to keep it open, that we'd get a better view in experiments. It's open so rarely in experiments, we don't really know what that structure looks like. And so it might help us do the experiments if we had a tool. It also might help antibodies because it would keep the spike open more often and potentially help antibodies bind better. But the one we're really most interested in is if we could prop this RBD open the same way that ACE2 does, would it fall off the way it does when ACE2 binds? Would it help that process that we already know is there a little bit? Could we trigger diffusing of these viral particles by making them lose all their S1 subunits? We don't know right now because we don't have tools to do that, but that's something that could be worth exploring. And so that's what we're going to try to do. So here's a simulation where our spike is opening, that RBD is moving up into the up position, and now you can see here, I've got this arrow pointed to a little tiny pocket where through the blue of one of our monomers, you can see a little pink behind it. That means that there's an opening. So what I'm going to do is I'm going to zoom around onto the inside of the spike and say, what's going on in that opening? What does it look like? Is there something we could do there? And so moving past all those glycans, moving to everything else, now switching to a wireframe, you can see that there's a hole there, right? And now I'm going to close that receptor binding domain back down, and you can watch that hole in this entire pocket disappear. That isn't there in the closed state. And so our thought is that if we could get a small molecule that would bind into that hole and stay there very tightly, that RBD wouldn't be able to close and it wouldn't be able to hide from the immune system anymore. And since the virus has been mutating itself to try to hide, we can counteract that. This is just a little bit better view of just this region of the spike that I've cut out, showing you where this pocket opens, and then when the RBD closes back down again, that collapses. This pocket is much better conserved. This is the same exactly the same in SARS, even though the antibody binding regions are different. This one is not, and it's because this is so crucial, it forms a hinge. This is like the hinge in the door, and this virus is not changing its hinges. This is just some gene sequence comparing that I won't go through for time. And since my movie worked, I'll skip those. That was just in case my movie worked. So you can say, well, why didn't we already know this from experiments? If there's a hinge there, we should have seen it. And it turns out that the experiments aren't good at these flexible spots. Here is one of the spikes in an experiment, and that's the hinge region that we're looking at. But there's quite a bit of the protein that's just too fuzzy, and we don't know what it looks like. Here's a better structure. We have a really high quality structure from X-ray crystallography, but it's only on the piece of the spike bound to ACE2, and our molecule would bind down into there, and that's not in the experiments. And so we don't have a good view of what this looks like in experiments. So here's a case where the computer models may help give us some good ideas about what to do. And so again, this is showing you the open or the up state has this vulnerable pocket where all of these amino acids are conserved, and the down state, that pocket is collapsed. We're going to try to wedge something in here to keep it from shutting the door. And Rob Rizzo's group, who's in applied math at Stony Brook, and his student Chris Corbo did some screening where they take large libraries of compounds and on the computer try to find ones that actually will fit into that pocket. And here's one that they came up with for us. They came up with a few of them, but they said, this looks good on the computer. It seems to fit really well. And then we're able to take that and do simulations. And so here's simulations, like I showed you before, of just that section of the spike with nothing bound to it. And it opens and closes, but it really likes staying in the closed state. And now I'm going to show you what it looks like when we have simulations and that small molecule bind, and that molecule stays bound to it, but it's unable to close, and the spike stays vulnerable and exposed during the whole simulation. So we've done that a few times and get the same effect. And so that's what we're exploring now for this part of our process of the spike function, and have experimental colleagues that are looking at molecules like this to see if we can test this in experiments and see if they actually do help keep the spike open. This is the surface that I showed you before, where it was only open about 1% of the time, and our simulations suggest now with that molecule bind, it should be open 99% of the time. We're able to shift this quite a bit over and keep this spike in a vulnerable state. Okay, so what happens later on in the process? This is where, to me, things get very interesting because the structure changes get to be so large. Here's our spike that I showed you before, and I'm just showing you the S2 part. This is after ACE2 is bound and after the S1 part has fallen off, hopefully induced by our small molecules. And now what has to happen? This is our pre-fusion structure. Here's the post-fusion structure, and it's hard to even see how these are the same protein. Hopefully you can see that they're quite different. This part down here in the bottom in blue has to come way up to the top. This part here in white is kind of bent over. It has to fold up, and we get a very different structure. So we spent a lot of time looking for pieces in common between these two and are able to overlap them. Not much looks the same. The green is the post-fusion structure and the colored one is pre-fusion. But we can zoom in and say, well, based on this, let's try to come up with an idea of how all of these things happen and what's the sequence of events in this membrane fusion process. So we have something, I won't go into the details of all of this because it's a lot of structural biology, but what it let us do was to go through and build a series of models. Here we have the one I've been showing you already, our pre-fusion spike structure. Here we haven't done a lot of building, just building in the glycans and then understanding more about the dynamics and vulnerable pockets and things like that. Next, we have models of what it looks like after that S1 subunit has been shed or dissociated. And we can say, what's going on here? How does that process happen? And then what happens to it next? Then, this is the one that I find very interesting. We have an intermediate model. This is unlike anything that's been seen in the experiments, where on the bottom it would be attached to the viral membrane. And then at the top, we've extended these helices that were folded up inside way up to insert these fusion peptides into the host cell surface. And that's the part that attaches the virus to the host cell. And so now that we have a model for that, we can start to ask questions about vulnerabilities there. And then finally, we have a post-fusion model, which again is very similar to what's seen in the experiment, but we've been able to build out more of the model and add in the glycans. One of the things that's interesting when you look at all of these, the green are these glycans that give you the shield. I had them blue earlier, but then I've got blue bits in my protein. So the green are the shield. And if you look at all of these states, the spike is really good at keeping itself mostly shielded all the time. It's the same set of glycans and they're attached in the same place. It's just every time it rearranges its structure, it shifts the glycans to a place where they shield it again, except for one. And this intermediate structure, where it's unfolded part of it to attach to the host cell membrane has a very large section of the spike that's not shielded and is very vulnerable. And so we think that that could be a good model for things like vaccine design or even small molecule or biological molecule inhibitors, because there's nothing shielding it in this state. But we have more work to do on this model. So that model comes in here. This is a really nice little cartoon from a paper a few years ago on coronaviruses. And that model corresponds to this one here, where after these S1 subunits come off, here's our S2, and it undergoes this change to extend and now attach itself to the host cell membrane. And so that's what we try to build a real atomic physics-based model for. And the next thing that happens is that it's thought that it's going to start to bend over, and that bending over motion eventually pulls those two membranes close enough that they fuse and the virus gets in. So now that we have a model for that state, we can start asking questions about how that bending over motion happens. So here's our model, the viral membrane on the left, the host membrane on the right. We haven't modeled them in yet. We're just in very early stages of exploring this, but we can say that it would have a refolding motion. And we can see that when it refolds, it's going to pull the section that's attached to the host cell membrane over, and it will be able to bring those two membranes in close proximity and induce fusion. And when we understand that process, how that works, we hope that that will give us a better idea of how to interfere with it. So we're not there yet. We have a lot to do. One of the things that we need is we need membranes. If we're going to look at things like membrane fusion and how it gets into the membrane, we need to have those membranes there. So we're building those in. Here's our spike molecule with our glycans on the surface built into models for the viral membrane. And then next, we need to build our models out so that they include the host cell membrane. And we're just in the earliest stages of doing this, where now we've got our ACE2 receptor in detail. There's a membrane here. This is just an early model of it. And we're going to be able to model this interaction and then understand how the spike will extend itself to insert here and pull those two together. And then eventually, working with Romy and Maro's lab, we're hoping to be able to do these simulations on the full virus. Who is just finishing up some model building on the full virus. And then we'll be able to take everything we've learned about the individual spikes and put them in a context of a virus and a more accurate environment. Okay, so what do we learn through all this computer time and using up lots of electricity and air conditioning? Well, we've seen that even though those experimental structures don't show us those glycans, the sugars that are on the surface, they're very important. They're covering almost the entire surface. They move around a lot at the pre-fusion state and at the post-fusion states, the two we've seen in the experiments, but it looks like the intermediate states might be vulnerable, and that could be helpful for us. We know that the immune shielding, its ability to hide from the immune system, is much stronger in SARS-CoV-2 than it was with SARS, and that's one of the reasons why we're having a harder time with this pandemic, is that our immune system is having a more difficult time with it, one of many reasons. But we found that there are pockets on the surface, like the one I showed you, that might help us control that and prevent it from hiding. And even though this virus is doing a better job, we may be able to fight back and help our immune system out. And then finally, I think that we're starting to make progress on understanding this mechanism, not just a structure-based drug discovery process, but a mechanism-based drug discovery process where we're getting the view of the cars, not just sitting in the parking lot or sitting on the LIE, but we're getting the view of the cars as they're going through their driving process, and as we're going through having the driver get in and out and opening doors and turning keys. And once we see how these pieces move, we hope that that's going to give us something that would be a treatment that would work not just for current coronaviruses, but the next round of coronaviruses, so we'll be better prepared next time. Okay, I want to thank first all of my team for doing all of these simulations. This is, those of you who've seen scientific talks before know that it's kind of obligatory to have a team picture at the end, and this is the pandemic version of the Zoom team pic. This is what it looks like when we're all meeting and talking about these things and working on our data. So this is my lab on Zoom in one of our lab meetings. Lucy Fallon did most of the work on this project that I showed you, has been doing all of these large-scale simulations. Lauren in the lab did a lot of work on the glycan model building and energy parameters for the glycans. Kellen has been working on these free energy surfaces to try to understand how they change when we have things bound. Yu Zhang has been working on the building things like the intermediate structures and these models that we don't know from experiments. Dasha has been looking at that S2 subunit and trying to understand how the fusion peptide gets out. Kenneth and Andre have been helping out with all of this too, and we have quite a few undergraduates in the lab that also have been helping on this project. And then I want to thank my collaborators. I mentioned Romy Amaro earlier. We've been working with her a lot. Arvind and Vigeli and Ronit. Ronit is an experimentalist that's working on some validation of those small molecules for us. And Rob Rizzo did the docking calculations. And we have lots of funding for the project, but also the COVID-19 HPC consortium. We would not be able to take on a project of this scale without all of the national supercomputer centers coming together and putting time towards these COVID-19 projects and giving us the level of supercomputer time that we've never seen before. And if you want more information about what we do, you can visit our lab website or contact us by email or ask questions now. And thanks for your attention.
Okay, thank you so much for Dr. Simmerling for this intriguing talk. So we should give him applause again. Yeah, thanks a lot. And I think there's some question coming in through the chat. So Christine will help to read some of them, right?
Yeah, we've got some really great questions here. One from an earlier part of your talk from Robert Molt. Is the fact that the domains are highly variable what explains why many such viruses in the family do not infect humans?
Well, I think yes. So SARS and SARS-CoV-2 both bind to the ACE2 receptor, and they have very similar structure and gene sequences in the part that's going to bind to ACE2. Other coronaviruses bind to other molecules, and some of them are molecules that humans don't have on our surfaces. And so they may bind to them, but we wouldn't get infected with them because they can't interact with molecules on our cell surface in a way that would unlock entry into the cell. And it's really about what interaction can trigger that conformational change in the spike domain. So I'm not sure if that's exactly what you're asking about, but the variability in the surface, much of it is that it's variable because it doesn't serve any function other than being on the surface. And some of it is variable because it's specific for specific receptors on the surfaces. And that's what makes them infect different organisms and different cells, is those changes.
Okay, great. Here's another one. This is a series of questions. I'll read the whole thing and then let me know if you want me to repeat anything. He says, I am curious about the scale of energetics of interaction between a spike protein and ACE2. I assume it is purely intermolecular forces, mostly hydrogen bonding. Is the network of intermolecular forces binding the two units together the result of 10 amino acids pulling together 100 amino acids? How global versus local are the conformation changes resultant from the binding of the units? How does this affect how many microseconds or milliseconds, gasp, are needed to achieve simulation of such a membrane-bound structure?
Okay, so these are good biophysicist questions from somebody who knows what they're doing. So if I can kind of paraphrase these a little for other people in the audience, this is a really big protein in the spike, and ACE2 is really large also. You saw that picture where I showed the two, and the interface between them is pretty small. And so the question is really about, what's causing this interaction and why is it favorable and why is it actually a really strong interaction when it's a small part of the surface? So on the scale, it is about a dozen amino acids that are at the surface. It's not very large as protein-protein interfaces go. It's a lot of hydrogen bonding. It's a pretty polar interface. There are some aromatic residues, but there's lots of charged residues too. So you can take that interface, since we know in detail what that interface looks like from crystallography, and you can come up with molecules that will mimic that interface. And that's been done recently. People like David Baker and some others have had really nice designs of proteins and peptides that will interact with that receptor binding domain very similarly the way that ACE2 does, which is not exactly what antibodies do. But you can design a molecule that will interact in very much the same way that ACE2 does and will compete for that spike with ACE2. And so it'll bind to your other molecule instead of binding to ACE2. And that's possible to do because the interface is pretty small. And so that's possible. How global versus local are the changes? There's not a lot of changes in the receptor binding domain itself. It's hard to know for sure because the receptor binding domain, when it's open and not contacting the ACE2, it's pretty flexible. And so we don't know exactly what it looks like. But in our simulations, it looks pretty similar to what it looks like when it's bound to ACE2. If there were a large structure change, that would probably give you a reorganization energy and it would weaken the interface. And so it's pretty pre-organized to bind to ACE2 as it should be. And so there's not a lot of conformational change other than some flexibility. And then the last one, how does it affect how many microseconds or milliseconds gasped, because somebody knows that we can't simulate that long or need to achieve such a simulation? Well, again, answering you as a biophysicist, we're doing a lot of these simulations with just pure MD, but those interactions where we form, I didn't show one, but we have that same thing where the spike opens and forms an interaction with ACE2. Those are not done just by brute force MD. We do things where we take the MD simulations of the endpoint structures, equilibrate them, and then map out pathways in multidimensional space, and then come up with progress variables and do free energy calculations and umbrella sampling and stuff like that, that you'll know all of these things that I'm talking about. But it's a very slow process. This is not something that we can just put the virus in, put the ACE2 in, and say, let it go, and it will go and do its thing like it would in the real world. And that's one of the things I didn't mention, that even using these largest supercomputers in the world, we can't model this directly. We need to use lots of algorithm tricks, and we need to poke and prod things. And one of the nice things about simulations is that we try to model physics as well as we can, but we also have the ability to go in and pull on stuff and push on stuff. And so we can ask questions that would be very hard for an experiment to go in some tweezers and say, well, what happens if I tug on this part? We can do that on the computer. And some of that is what helps us model things that are just beyond our ability to just wait for them to happen on their own.
Okay, great. Another question here from Christopher Janata. What force field is used for these simulations to be able to calculate all these interactions for microseconds long?
Must be another biophysicist to talk about force fields. So the force field is, another word for that is an energy function. All of these simulations are guided by energy. And what we do is we try to make our energy, which gives us forces that tell us how molecules are moving and how all the atoms are getting pushed around. Those energy functions we train based on physics. And so the force field we use from a technical point of view is called FF99SB, only SC. It's one that's been developed in my lab and many people around the world use. It's available in most simulation programs. It's the one that was used for all those studies I showed you in the beginning for validation. And we train that based on much smaller systems where we do much more expensive physics calculations, quantum mechanics calculations, to look at the energies in a way that's hopefully very accurate. And then we go through a computer training and train our models that we use in our bigger system. But that's the force field that we use. And there are limitations. We know we could do a better job, but then it makes the calculations slower and it makes the systems too hard. So we're always trying to compromise between how accurate you are and how much biology you can simulate. And that's why computers getting faster every year is really good for this field.
Okay, great. Another one here from Robert Molt. Does your choice in steering the reaction coordinate for this significantly affect the results? I know this is a tough question, but your commentary is nonetheless insightful.
So this relates back to what I said earlier about how we can't just put this molecule in and have it do its thing on its own, that we do need to steer it a little bit. We need to guide it. And so the particular guiding that we use, we can take the closed structure from the experiment and the open structure from the experiment, or we can take a model that we built or something like that. And then we use a method called nudged elastic band that maps out that pathway, and we don't pick a coordinate. It just does it in the full Cartesian coordinate space. Then in order to do the analysis and be able to plot things for you, like free energy, we do have to pick a couple of things. You know, there are 2 million atoms in the system and they all have XYZ coordinates. And so you can vary any of them and get changes. And so we have to plot it in a way that helps us understand what's happening. And the way we do that, at first, the graphs I showed you are really just from looking at structures and trying to use our insight as structure biologists. But then I showed in my list at the end of our collaborators, Arvind Ramanathan, Arvind's group is using artificial intelligence, and he takes our simulations and they do an AI-based, like a deep learning approach to try to reduce the dimensionality of the system and come up with what would be appropriate reaction coordinates. And so that's what we're starting to use now, because it is very difficult to pick those out, but we don't need them in the beginning and the steering part. We only need it during the analysis part. All
right, great. More good questions coming in from Vladimir Koropin. are quantum mechanical effects important for this?
Well, so again, physics people. So this is a classical model, not a quantum mechanical model. So classical model meaning we have particles that have masses and our bonds are attached together by springs. We have a lot of limitations in the model. It's faster than quantum mechanics, but we can't model chemical reactions, for example. And so if we wanted to look at how a reaction would happen, how you break or form a bond, we can't do that. And that's definitely a quantum mechanical effect. There are other quantum mechanical effects that are not as obvious that we do try to model in our energy functions and our force fields. For example, rotation around double bonds, for those of you that are chemists. It's very hard to rotate around a double bond because there are pi orbitals that you break. It gives you a high energy barrier. And so in our energy function, we have trained it for things like aromatic rings and double bond rotation. And we've trained for lots of orbital effects. But because they go into our training, they're static. And one of the things that we can't do is have quantum mechanical effects that change during the simulation. That's not something that we're at yet. And so I think for these simulations that they're very dominated by classical effects, intermolecular interactions, salvation, hydrophobic effect, hydrogen bonding, all of these things. And I think we do a reasonable job of those. But there are certain things definitely that this would not be a good choice for, and chemical reactions is the obvious one.
Thank you.
So Dr. Simmerling, there are two more questions. Are you able to help answering it before?
Sure, I'm open as long as we have time here in the session.
Sure, yeah, thank you.
Okay, yeah, we've got three more here. Here's a quick one about one of the cartoons. Is the action of the spikes cooperative with one another or independent of one another? Your cartoon implies just one point of contact is needed for cellular assimilation. Am I overinterpreting the cartoon?
So the answer is I don't know, and I'm not sure that people know yet. Most of the structures that we had are bound to a single ACE2 because we have to cut the spike apart to get good structures. But now as people are doing better and better cryo-EM structures and even cryo-electron tomography where we have those entire viral particles, we're starting to see spikes bound to more than one ACE2. It's probably not clear yet what happens. From our simulations, it suggests that when one of the RBDs open, that the other ones will be easier, but other than seeing why that would be the case, we don't have actual data to support that yet. And I'm not sure that we know from experiments other than usually only seeing one open, and only in rare cases do we see more than one open. If they were cooperative, you would think you would see none open or you would see several open, and that's not true. But what might happen is when you open one, it may be easier to open the second one than to open the first one, but it definitely doesn't make all of them open.
Okay, we've got two more. Does inserting the small molecule into the gap of the RBD have an effect on binding probability to ACE2, other than just being open more often?
Well, so I don't know, again, and we don't know, and we certainly don't know experimentally because we haven't done these experiments yet, we're just starting them. But I would guess that it would make it bind more tightly to ACE2 because when it's in the bound form and covered by those glycans, it can't bind to ACE2. And so if we prop it up and make it more exposed, then overall, it will help it bind to ACE2 because it doesn't need to escape from the glycan shield. But it depends a little on the perspective that you look at. The RBD will bind to the ACE2 the same way that it did before, but overall, since it's not hidden as much, the ACE2 might be able to capture it more, and it's about shifting equilibrium. And so it may make it bind to ACE2 better, but there are experiments that show already that the affinity of binding to ACE2 is not a limiting factor in the infectivity. So even though it may bind better to ACE2, it would not make it have an easier time getting into the cells, we think, but we don't know yet. We're hopeful that binding that molecule there will make it more vulnerable before it gets to the ACE2 and that this question won't be as relevant, but we're not sure yet.
Okay, great. Last one. Referring back to the theorized intermediate structure with the exposed extended helices. Did I pronounce that right? Mm-hmm. Within the exposed extended helices, have similar extension mechanisms been studied in viruses in the past?
Yes, and did not mean to imply that that mechanism of the extension and the way it inserts is something that wasn't known. That is something that is fairly well accepted, that that's what happens. What we didn't have is an atomic detail physics-based model for it, at least not for coronaviruses that I'm aware of. But that extension of those helices and insertion of fusion peptides is not just a coronavirus mechanism. That's something that happens in other viruses. And one classic example of it is HIV. That HIV has a different protein. It doesn't have a spike on its surface, but it does have a viral fusion protein. And it's thought to work in a very similar way, where it extends these helices, inserts into the cell surface, and then there's a rearrangement that brings about cell fusion, even though it's a different protein. And that is actually being studied by Rob Rizzo's group in applied math at Stony Brook, where they are looking for inhibitors in the case of HIV, possible AIDS treatment that would come from blocking that intermediate structure from progressing on to the next step. So I think we have an idea of generally what that looks like. What we don't know is specifically what it looks like, and we don't have enough information that we could start doing any design work based on it, and so that's what we're trying to provide.
Okay, I think that's all our questions.
Okay, thank you everybody for taking time out of your day to listen to me. I appreciate it.
Thank you so much again, Dr. Simmerling, and thank you for this very engaging conversation. Really appreciate it. Thanks, everyone. Yeah. So you all have a good day.
Thank you.
Thank you.
Yeah.
Thank you.
Yeah.
Thank
so
you.
much. Yeah.
Thank you.